Hi, my name is Leonardo Ancheta, and I am representing Advanced Targeting Systems located here in Carlsbad, California in the United States. And I'm really thankful for this opportunity to be able to speak to you about some research tools that we really think is going to be helpful in that space regarding the research into dementia, Alzheimer's disease, Parkinson's disease, and just other neurodegenerative diseases in general. And just really trying to make progress in meeting the needs or at least trying to slow these diseases down. And so the title of the presentation today is Saporin as the Scalpel in Molecular Surgery to Create Models in a Way that Can Mimic Diseases. And so I'm going to try to spend the rest of this presentation in trying to explain what Saporin is, how it's currently being used, and hopefully at the end of the presentation you'll be able to see why this might be a research tool that will be helpful in your lab. All right, so let's get started. As an overview for the presentation, the first thing we'll do is just talk about what Saporin is in general, and we'll talk about the mechanism of action and how it works. Then we'll follow up with the actual different disease models that Saporin conjugates work in. And so we'll talk about Alzheimer's disease, Parkinson's disease, narcolepsy, amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. We'll talk about temporal lobe epilepsy, and then we'll finish it up with a summary of Saporin conjugate disease models. All right, so the first thing on the list was a mechanism of action for Saporin, all right? And so first off, Saporin, what is it? So Saporin is a 30 kilodalton protein isolated from the plant Saponaria officinalis. We actually still isolate and purify Saporin from the seeds, but we also have the capability of making it recombinantly. And so this protein is part of a family of the ribosome inactivating proteins, also known as RIPs. Popular RIPs also are like ricin or abram, which you may have heard of. And so the way that Saporin works is that it cleaves an adenosine at the N-glycosylic bond on the ribose backbone of ribosomal RNA. And so Saporin becomes a functional research tool because Saporin lacks a B-chain lectin. So essentially it can't bind and enter a cell on its own. So the functionalization of Saporin actually is only made possible by attaching a targeting agent to it. So for example, like an antibody or a peptide, growth factor, or even an aptamer. So anything that can bind to a cell surface target and then get internalized could be used as a targeting agent, all right? And so we'll just kind of run through the mechanism of action really quick. So we'll take an example of an antibody conjugated to Saporin, where the antibody binds to its cell surface target. Everything gets internalized into the cell through receptor-mediated endocytosis. Once inside the cell, the Saporin's cleaved from the antibody, leaves the endosome where it's free to target the ribosomes. And then once it targets the ribosomes, and then activates them, and that eventually results in cell death. Okay, so now we'll talk about one of the first disease models, and that's going to be Alzheimer's disease. And we create an Alzheimer's disease model using a product called 192 IgG sap, and it targets the low affinity neurotrophin receptor, otherwise known as P75. And so 192 IgG sap is a monoclonal antibody that targets P75, recognizes the low affinity neurotrophin receptor, and we've chemically conjugated it to Saporin. And so we've created this immunotoxin that can permanently eliminate the cholinergic neurons in the basal forebrain, but also in the medial septum and the diagonal band of Broca, which provide critical inputs into the hippocampus. Just to take a minute though, I want to emphasize that the amount of cell death that you see with a Saporin conjugate correlates with the number of cells that express that particular receptor, right? It's not the amount of Saporin attached to the targeting agent. It's really the amount of expression of that target receptor, all right? And so right now we'll take a look at an example. Again, this is ablation of cells expressing low affinity neurotrophin receptor P75. And right now we're looking at this tissue taken from rats injected with 192 IgG sap. And so panel A is going to be the control, And then panel B is going to be the treated sample. And then this was intraventricular injection of 192 IgG sap. And you can see in panel B almost the complete loss of those P75 positive neurons. And what's nice though is you'll notice that you're not actually seeing a lot of physical damage, like with like a chemical or like something burning the actual tissue to remove that. You're actually just seeing specific loss of those particular neurons. All right, and in this next slide here, in this next example, again, we're just looking at more examples of using 192 IgG sap, and this is crescent violet staining of the medial septum region after ICV injection of 192 IgG sap. Again, so panel C will be the control, panel D is the treated sample, panel E will be control, and panel F will be the treated sample. And again, what you're seeing here is just the significant loss of those particular neurons that were targeted within 192 IgG sap. And again, you're not seeing like actual physical damage to the tissue, just the loss of those particular neurons. And so by lesioning these neurons of healthy laboratory animals, you're essentially creating a representative model of the symptoms of memory deficit associated with Alzheimer's disease, right? So you're not just looking at, okay, these neurons are disappearing, but it's actually creating the symptoms of Alzheimer's disease in the rat. Okay, the next model that we'll review is the model for Parkinson's disease. And for this disease model, we can target and lesion dopaminergic neurons with antibodies directed against the dopamine transporter as well as dopamine beta hydroxylase. And so we're targeting both of those. And the tools we have are called anti-DAT sap or anti-DBH sap. And these both use monoclonal antibodies that have been attached to Saporin. Okay, and so with Parkinson's models, a common way that researchers have attempted to replicate this is to use the neurotoxin 6-hydroxydopamine, also known as 6-OHDA, and that selectively destroys dopaminergic and noradrenergic neurons in the brain and can induce Parkinson's symptoms in laboratory animals. And yes, this is a valuable tool, but there have been a few issues with this model. And hopefully this is where, a Saporin conjugate can be helpful. So, for example, some issues with 6-OHDA include not producing the extranigral pathology or the Lewy body inclusions. And so lesioning with a Saporin conjugate is also helpful because it's going to be more specific than 6-OHDA because the lesioning only occurs in places essentially that express either the dopamine transporter or dopamine beta hydroxylase, right? And so we're going to be looking at this example here, and this image is taken from Wiley et al. 2003, and these are from rats treated with a conjugate targeting the dopamine transporter, so that's anti-DAT sap. So panels B, D, and F are going to be the controls, and panels A, C, and E are the panels where they receive treatment. And so again, you're going to notice the loss of the dopaminergic neurons for the substantia nigra, pars compacta. You'll notice these little arrowheads here pointing to those areas. And you'll notice that the greatest loss was seen from panel A, and that's because it received the highest dose of anti-DAT sap at 2.8 micrograms. Again, this is a good representation of a Parkinson's disease model. Again, we're not just looking at removing these particular neurons in histology, but we're trying to create the symptoms as well. So you're looking not just studying tissue, but you're looking at the behavioral facets of the disease. The next disease model we'll cover and review is for the disease model of narcolepsy. And we create this using the orexin peptide conjugated to Saporin, right? And so this is the ablation of histaminergic neurons, right? And so just in general, the orexin A and orexin B receptors, also known as hypocretin 1 and hypocretin 2, are found in the parafornical area / latero-posterior hypothalamus, all right? And projections from this area cover much of the brain. And so these receptors have been implicated in various neurophysiological and neuropsychological disorders such as narcolepsy, insomnia, drug addiction, anxiety, and even migraine headaches, right? And so what we've done is we've taken a orexin peptide that is rat/mouse, and we've created a orexin B sap conjugate where we've chemically conjugated them together with saccharin. And so we'll take a look at an example. And the image here is taken from Geraschenko et al. 2001, and this is tissue taken from rats injected in the lateral hypothalamus. And what you're going to see here is these results basically resulted in rats having narcoleptic symptoms and would fall directly into REM sleep, while 1 second just happily eating earlier, right? And so in the tissue here, you're going to notice that asterisk right there is the site of injection, but you're going to notice the marked neuronal loss there. Again, you're not seeing a lot of physical damage to the tissue, but just really specific neuron loss. So next on the list is the disease model for amyotrophic lateral sclerosis, otherwise known as Lou Gehrig's disease. And this model is created where we deplete motor neurons expressing the GM1 receptor. And we do this by using cholera-toxin B chain that we've chemically linked to saccharin. Okay, and so just as an overview, amyotrophic lateral sclerosis, again, Lou Gehrig's disease, is a disease that leads to degeneration of motor neurons and skeletal muscles. And that includes the muscles required for swallowing. And so tongue weakness is one of the earliest signs of bulbar dysfunction in ALS, and that's attributed to the degeneration of motor neurons in the hypoglossal nucleus in the brain stem, and the axons of which directly innervate the tongue, right? And so dysphagia, also basically is difficulty swallowing. So dysphagia and strategies to preserve swallowing function have rarely been studied in ALS models. And it's difficult to study dysphagia in these models because the amount and the rate at which motor neuron death occurs can't really be controlled. And degeneration is not really limited to the hypoglossal nucleus anyways. And so what we're going to try to do is a lot of the work that is out there right now is using intralingual injections of cholera-toxin B conjugated to saccharin to study the impact of hypoglossal motor neuron death with many of the complications that are present in ALS models, all right? And so the example we're going to be looking at right now is taken from Lind et al. 2018. This is tissue taken from rats treated with CTB saccharin. And essentially what you're going to look at here are panels B and C are going to be control, and panels E and F are going to be the treated sample. And you're going to notice a lot of these little arrows here, and those will be essentially pointing at the dying or dead neurons that were affected by CTB. Again, so you can just see the marked loss, not a lot of physical damage to the tissue, but just specific neuronal loss because of the targeting agent. So the last disease model that we'll cover in this presentation is going to be temporal lobe epilepsy. And we accomplish this by lesioning the GABA inhibitory interneurons via a conjugate called stable substance P saccharin. And essentially that's substance P peptide that's been chemically linked to saccharin. And so just as a general, temporal lobe epilepsy with hippocampal sclerosis is a common neurological disorder characterized by seizures that arise from the hippocampus and related structures, often as a consequence of brain injury. Right? And so the idea was that disruption and inhibition from injury-induced neuron loss or GABA neuron dysfunction might be sufficient to cause temporal lobe epilepsy with hippocampal sclerosis. And so the publications out there right now using this model, they target hippocampal GABA neurons along the longitudinal expanse of the hippocampus in the absence of any other brain-induced injury, and to see if that was sufficient to initiate hippocampal eleptogenesis. And so receptor-mediated lesioning with substance PSAP was highly selective because the neurotoxin enters the GABA neurons via the NK1R receptor, and that happens to be what all hippocampal GABA neurons also express, the NK1 receptor. So that was nice. And the authors go on to describe how targeted hippocampal GABA neuron ablation produced hippocampal sclerosis, epilepsy, and the dissociable effects on the Morris water maze and object place paired association tasks. And so we'll take a look at this example. And this image was taken from Bumanglog et al. 2019. And this is rat tissue taken from rats treated with substance P. And the interesting point here is that this was 1 year after treatment with the substance P. And so again, this is showcasing the idea of permanent removal of a particular set of neurons. Again, so this is about 1 year after treatment. And basically, you're seeing here with panel 1 being the control, and panels 2, 3, and 4 are the tissues that receive treatment. And again, you can see the loss of neurons in the sections with the dentate gyrus, CA3 and CA1. You can see the particular specific neuron loss there. Again, not a lot of physical damage to the tissue, but just again, you're seeing specific neuronal loss without damaging or targeting other neurons. Again, this was just a nice example of a permanent removal of those neurons. Okay, so I would just like to finish the presentation with this chart. And I think this is a nice chart that highlights the idea that, Saporin conjugates, it's really a platform that's really open-ended, and how it can be used as a research tool. So this chart basically is a representation of taking like over 200 articles, right? And they all are using Saporin conjugates to create animal models or diseases. And then we broke them up into categories. And so the largest subsets that you can see were ones I just discussed, like dementia, Alzheimer's disease, and memory being the top tier. But as you can see, though, with this chart, we're not just looking at neurodegenerative diseases, but we're looking at topics of pain and itch. And so again, this is a nice example of just being able to show that Saporin conjugates, there is really no limiting factor. It's just how creative you can be and how it can be used, right? And so again, I just want to thank you for your time, and I hope if there are any questions, you just reach out to me, and I would love to hear if you have an idea of how you think Saporin conjugates can be used in your lab, and I would love to speak to you and see if I can help you out. Thank you for your time.